I am Ruxandra Dafinca, and I'm a senior research fellow at the University of Oxford. My background is in molecular biology. So I'm a molecular biologist working in neurodegeneration, specifically in iPS-derived models of disease. I'm obviously very thankful for the opportunity to have this grant and to start off with this project. The project that has been funded by the Catalyst Award is aiming to answer the question, what are the local molecular dysfunctions that occur in the pre-synapses of ALS motor neurons that then lead to loss of synaptic transmission? In order to do this, we will be using a combination of novel spatial proteomic techniques, bioengineering tools, as well as computational biology, so machine learning. And we know that maintaining neuronal health relies on proteins being in the correct place in order to fulfill their function. And we also know that in motor neuron disease, we have the mislocalization of TDP43 in 97% of the cases, which highlights the critical importance of protein organization and localization within the cell, not just its expression levels. So we are, however, lacking a clear, comprehensive view on what other proteins are doing at the same time, and where other proteins may be what other proteins may be mislocalized that could then become key targets also for developing new treatments. And intervention. So in this project, we will use the novel spatial proteomic techniques with the bioengineering tools and with computational biology in order to determine the precise location of all the proteins that are located in the axons, which may be driving neuronal death in MND. The presynapse is the area of the neuron where communication is being prepared. So the messages that get transmitted between neurons, which is how they communicate with each other, are being prepared in what we call the presynapse. So before the synapse, which is the space in which the messages are being transmitted. So these messages need to be packaged. The right proteins need to go in there so that they're transmitted to the next neuron to perform its function. And we know that this communication breaks down in motor neuron disease. But what we don't know is at a higher resolution than we've seen before, what happens in this space before this breakdown occurs. And this is what this new technology that we're importing now from biochemistry, looking at high resolution spatial proteomics at all of the proteins that are located within this area. What are they doing? Where do they localize? And how are they organized? And how are they different in a neuron that is affected by motor neuron disease compared to a healthy neuron? Funding in basic mechanisms and in understanding motor neuron disease at the cellular level is really critical in order for us to be able to develop more effective treatments. And there have been so many advances now in proteomics, in transcriptomics, in computational and machine learning that we can now integrate into motor neuron disease research to be able to understand at an unprecedented level of detail how the molecular dynamics are occurring and what dysfunctions are occurring in the neurons affected by the disease. And with these current innovations, we are therefore in a position to really understand local perturbations at this point. And this is a pivot, like a really important aspect, I think, because motor neurons are so long, they have long axons, they have this requirement for local subcellular regulation and local transcription, local proteomics, everything needs to be in the right place at the right time. And we have the tools to now look at these differences, at what happens in every part of the neuron. And understanding this, I think, will give us really a lot of insights into the mechanisms that are driving loss of synaptic communication and potentially a lot of new targets that we can think of for downstream therapy. I hope that my research will be highlighting potentially new targets that can be therapeutically useful downstream. And I hope that I will also manage to build a comprehensive map of proteins and their organization locally in the axon and in the presynaptic compartment that can then help guide other research into different pathways and and to develop drugs for different pathways that may be affected that we haven't perhaps considered before using our previous technology available. This study will be the first to create a detailed map of the proteins and how their organization may be disturbed in motor neuron disease. And this will help us identify changes that may be leading or driving motor neuron death. Integrated view really will provide hopefully critical insights into the cellular processes that are affected and to understand better motor neuron death. As for the next steps, we will of course need to validate our findings, first of all, validating whether the proteins that we have found are actually relevant and their mislocalization is actually relevant to disease. So that would be the first step in terms of experimental steps that we will be taking. So validating that the mechanisms and pathways we are identifying are actually relevant to motor neuron disease. And further on from that, the pathways we do identify, new pathways identified, we can then collaborate with other drug screening platforms, for instance, the UK Motor Neuron Disease Research Institute. We would be looking at repurposing drugs and looking at the targets of those drugs that are currently being repurposed to see which of them could be affecting the pathways we've identified, and to test those in a more high throughput manner than we are currently doing in this project, in a more high throughput manner to understand whether we are improving, targeting those pathways would improve the neuronal health of the motor neurons. So while this is not a therapy development project, it will have an impact on understanding and bringing new targets, potential new targets for more effective treatments. So I think we will be identifying hopefully key pathways involved in neuronal death with a high resolution that hasn't been done before in motor neurons, and hopefully these will be essential for understanding the mechanisms of disease and also for prioritizing targets for future drug screenings. So I think this type of mechanistic understanding is really critical to enable further downstream rational drug discovery, even if this work itself is not directly therapeutic, or it is more the discovery phase of the whole pipeline.